Hello, everyone. Welcome to my video tutorials for spatial transcriptomics data analysis. In my previous video tutorial, I showed you how to perform spatial transcriptomics data deconvolution using the robust cell type decomposition package in R. So now is a Python package called cell2location. It has the same function to perform spatial transcriptomics data deconvolution. So in today's video tutorial, I'm going to show you how to use the cell2location package in Python. So here is the website for the cell2location package. If you use this package to analyze your data, you should set the cell2location paper in your publication. So I'm going to use the same dataset for the human lymph node spatial dataset generated by the 10 times genomics Visium technology. If we scroll down, you can see the workflow diagram. First, you need to prepare a reference dataset for the lymph node. Then you have your spatial dataset. In today's video tutorial, I'm going to show you how to prepare the reference data set and also the spatial data set. Later, we can use the cell2 location package to perform the spatial data deconvolution. So now we can go to Jupyter Lab. First, we can load the packages. You can see I installed the cell2 location tutorial packages. We also need ScanPy, NumPy, and Matplotlib to create the images. So for the analysis, we can generate a result folder, and in the result folder, we create a lymph node analysis folder to save the data. So first, we are going to estimate the reference cell type signatures using the negative binomial regression model from the cell2location package. If you want to use the same dataset for practice, you can download the lymph node reference dataset from this link. I downloaded already. We can just use the ScanPy function to read the data in as a data reference. Okay, we load the data. We can have a look at this dataset. You can see we have 73,260 cells in the dataset, and also we have 10,233 genes. You can see all the information in this dataset for the observations, variables, unstructured layers, PCA and UMAP in the observation multiple layers. So this is the reference dataset. We can have a look at the variables in the reference dataset. You can see for the variables, the row names are the gene names and the gene IDs in the first column, gene ID 2. Because later we need to use the same row names for the reference dataset and also the spatial dataset to subset the genes that are presented in both datasets. So we need to make some changes. We will set the gene ID as row names. We can set the row names, they are the gene names, as symbols. Then set the gene ID from the gene ID 2 column as the row names. At the same time, we will remove the gene ID column. Let's run. And we can have a look at the reference data variables again. You can see now the row names are the gene IDs. If we go to the end of the columns, you can see we add a symbol column to keep the gene names in the variables. So now we have the reference data set ready to perform permissive gene selection. This is a similar process Next, we identify the highly variable genes by the ScanPy package or the thread package. But for the cell2 location package, they prefer to identify the standard highly variable genes. Use the filter genes function from this package. You can see we are going to filter the genes, keep the cell count cutoff from 5, then cell percentage expression will be 3%, and the mean cutoff will be 1.12. So it is depends on your data set. You can change in those parameters. So first we will filter the genes, then we can subset the reference data set to keep the genes that match our criteria for filtration. Let's run. You can see we generated a plot to show the number of expressed genes against the expression levels. Normally we filled out the genes that are inside this yellow square because this reference data set was filtered in previous data analysis. Here in the rectangle square, we don't have any genes because those genes already been filtered out. So we selected the highly variable genes. Now we can set up the regression model for the reference data. You can see the data will be the A data reference. Batch key we set as samples, and the labels key as a subset. You can find all the information from the A data reference object. So let's set up the regression model. Now we can create the regression model for the A data reference. So we created the model. We can have a look at the model setup. Use model view A data setup function. You can see all the parameters we set up for the analysis. I'm not going into details. You can have a look at the cell2location package website. So we got the regression model ready. Now we can train the model to estimate the reference cell type signatures. Later we will use those signatures to label cell types in the spatial dataset. So I ran this model training before. It takes more than 4 hours. So I'm not going to run the model training in today's demonstration. You can run the code here to perform model training. You can see we set the max epochs as 250. It depends on your data set. You may have to increase this number to train your reference data set. Later we can see if 250 is enough for this data set. Use the model plot history function. So as I said, I'm not going to run the model training function here today, but you need to run it. At the end of the training, you will see the sentence that training reached to the maximum epochs as 250. So instead, I ran the model training before and saved the data in my result folder. After you run your model training, you can use the code here to save the data. We can go to my result folder and have a look. You can see in my result folder, we have the lymph node analysis, and also we have the reference signature. We can click and open the folder. You can see in the folder we have the model training for the reference data set, and also we saved the signatures as SCH5AD. So now we can just go back to Jupyter Lab and then save the model and the output. We can use the code here to load the result. Okay, we noted the result. You can see I have an error here to display the widget. This is because I installed the new software, and the software is not compatible with other softwares, but it doesn't affect my data analysis, so we can ignore the error here today. As I mentioned before, we can use the model plot history to determine if the model needs more training. Let's run the model plot history. It will generate a plot. You can see it is very similar to the plot we have in the thread package as elbow plot. We didn't show the first 20 ELBO nodes. From the model training history, you can see the ELBO nodes gradually decrease, but when it reaches to around 100, it starts to become flat, which means 250 training epochs are okay for this dataset. Okay, so that's the reference dataset. I didn't show you how to run the model training to save time for the video tutorial, but you can run it by yourself. So next we can prepare the spatial data. So for the cell2location package, it has a small demonstration data V1 human lymph node spatial data installed in the package. Once you install the cell2location package in Python, you will have this demonstration dataset. You don't need to download, but you need to find where the data it is. Depends on where you installed the package. So we can load this VM demonstration data as a data VM. Now we can have a look at this demonstration data. You can see we have 4,035 spots and 36,601 genes in this dataset. Again, we need to make some changes for the spatial dataset as we did for the reference dataset. First, we can add a sample column in the metadata to show the sample information for each spot. Let's create the sample column. Now we can have a look at the observations. You can see the row names are the cell barcode for each spot. Then we created a new column as a sample to save the sample information as the V1 human lymph node. Now we can have a look at the variables. You can see now the row names are gene symbols, and we have the gene IDs in the first column. We will do the same to keep the gene names as symbols in the variables. Then we set the gene IDs as row names. At the same time, we delete the gene ID columns. Let's have a look at the spatial data variables again. You can see now the row names are the gene IDs, and we have the gene names in the last column symbols. So now you can see we use the gene IDs for the reference dataset, and also we use the gene IDs for the spatial dataset. So later we can use the gene IDs to select the genes that are expressed in both reference and the spatial dataset. So we change the row names. Now we can remove the mitochondria genes because we are not using mitochondria genes for the spatial data analysis. If it's in human, will be capital letter MT hyphen for all the mitochondria genes. If you use mouse data, will be small letter MT hyphen. So we can find all the mitochondria genes from the spatial data variables. You know they are in the symbol column. We can find it now. So next we can remove the mitochondrial genes for the spatial data analysis. Okay, we prepare the reference data set as a data reference, and we also prepare the spatial data set as a data vision. Now we can do the spatial mapping. This will be the step to perform spatial data deconvolution using the cell2location package. So we did the module training for the reference dataset. Now we can extract the reference cell type signatures as a data frame for spatial mapping. So the code here shows you how to extract the reference cell type signatures from the trained reference dataset. We can have a look at the first 5 rows and 5 columns for the data frame. You can see the gene IDs for the first 5 rows. Then we have the estimated expression level for each gene in different cell types. So because the reference dataset and the spatial dataset were generated by different technology, so the genes that are presented in one dataset may not presented in the other dataset. So we can use the intersect function to select the genes that are presented in both datasets. Once we have the shared gene expression, then we can use the shared gene names to subset the spatial dataset and also subset the data frame for the reference gene expression. We can run it now. So we have the data ready to perform the spatial dataset cell type deconvolution. First, we need to set up the module again. Now you can see we are going to set up the A data as a spatial dataset. The batch key will be the samples. We can set up the module. Next, we can create the module for the module training. You can see now the data will be spatial data, but the cells data frame comes from the reference dataset. The other important argument here you need to set is the n cells per location. This is the total cell number in each spot. You should know it from your spatial dataset. So next will be the detection alpha value. You can have a look at the cell2location website. They explained in details how you choose the parameters for n cells per location and the detection alpha. You can see for today's demonstration, we are using 20 for this dataset, but they recommend you also try 200 for your dataset. So now we can create the training module. Use the code here. Let's run. Again, we can use the module view to see the parameters for module training. For the spatial mapping, we can run. You can see all the parameters here we set up for the module training. So next step is to run the module training again for the spatial mapping. You can see here the maximum epoch is 30,000. So I run this step before, and this step takes even longer on my computer. It took more than 24 hours to run this step. Again, I'm not going to show this step today. You can run on your computer. I use the code down here to save my previous analysis as SP-H5AD in my result folder. We can just use the code down here to load the save the model and the output. We can go to my result folder and have a look again. You can see I saved my model training in the cell2location mapping folder. You can see the data SP85AD. So now we just load the data for the demonstration. We can load it now. So we loaded the model training data. Because we use the maximum epochs 30,000, we can ignore the first 1,000 epochs and plot the model history again to see if we performed enough model training. Let's run. You can see from the plot, the ELBO nodes became flat after 10,000 training epochs. So the maximum epochs we used in the model training will be fine for the spatial data cell type deconvolution. So until this step, we already performed the lymph node spatial data cell type deconvolution using the reference dataset. Now we can have a look at the result. First, we can realize the cell abundance in spatial coordinators from the lymph node. We can use the 5% quantile posterior distribution to represent the confident cell abundance. So let's add the 5% quantile into the data. Then we can select one slide from the spatial data image to plot the cell abundance. Following settle all the parameters for the plotting, we can show the B cell and the T cell abundance in the lymph node spatial dataset. Let's run. Okay, you can see we generated 8 images to show the cell abundance for the cell types that we set up here. We have the B cell, T cell, and the sentinel cells. So here we show the cell types on individual image. We can also make a plot to show multiple cell types in one image. For example, here we are going to show the CD4 positive T cells, B naive cells, and dendritic cells on one lymph node slide. Let's run it again. You can see now we generated the image to show CD4 positive T cells, B naive cells, and dendritic cells. They are in yellow, brown, and blue color, respectively, on this image. Okay, you can see in this video tutorial, we followed the online tutorial to perform spatial transcriptomics dataset cell type deconvolution using the cell2location package. I'm going to stop from here for today's demonstration. If you want to use this package for spatial dataset deconvolution, you can install the cell2location package and try to run the lymph node dataset or your own dataset. Thank you for watching my video tutorials. See you next time.